Okay. Today, we're going to talk about the different stages of meiosis. Now, these shouldn't be terribly hard to learn because, like I said, they follow sort of the stages of mitosis. Right. There's 2 of each. We don't have anything. Ah. Alright, for some reason the markers aren't working, so I'm going to go at it this way. So, the first stage of meiosis is prophase 1. If you remember from yesterday, we're going to split meiosis up into 2 kind of sections. There's meiosis 1 and meiosis 2. And the first stage of meiosis 1 is prophase 1. Now, many of the same things happen in prophase 1 as happen in prophase of mitosis. The nucleus dissolves. The DNA condenses. And the spindle forms. This is all part of meiosis. Okay, but there's also something quite special that happens that makes meiosis 1 pretty distinct from prophase 1 and mitosis. And that's that the homologous chromosomes line up in tetrads. Okay, you guys ever play Tetris? Yeah. Kind of an old school game, but it's a classic, right? Anybody know why it's called Tetris? Because of that? No, not because of that. Any ideas? Nobody? So tetra means 4. If you ever play Tetris again, look, every one of the little shapes that comes down is made up of 4 boxes. Just the arrangement has changed. So that's why that's called Tetris. In this case, the tetrads are called that because there are 4 sister chromatids all kind of stuck together. Okay, and this did not happen in mitosis. In mitosis, each of the homologous chromosomes stayed separate from each other. But in meiosis, those homologous chromosomes come together and they stick together, and so you have 4 sister chromatids all kind of making one structure, and that structure we call the tetrad. And the reason why this is important is because there's a special thing that can happen in this case called crossing over. Okay, if we look at these tetrads here, these two sister chromatids, when they're lined up against along their length, the DNA that makes them up is kind of against each other. And what can happen is these can actually get swapped around, and that's what we call crossing over. So I've got a better picture of that on the next thing here. Now, we have to be careful here. We need to realize that there should be another sister chromatid here and another sister chromatid here. We're just showing the two that are next to each other just for some clarity. So during crossing over, the two sister chromatids, and these would have very similar genes on them, can actually switch, and a chunk of this can break off and switch places with a chunk of this. And so we end up with what we call recombinant DNA. So these two DNA strands, even though the genes on them are similar, they may have slightly different variants of the genes that we call alleles. So for example, this chromosome right here might have a gene for brown eyes on it, and this chromosome right here might have a gene for blue eyes on it. Now, whether you have blue eyes or brown eyes depends on which one of those is dominant. But those can actually get switched around with each other and end up on different sister chromatids than what they started. And if we compare those to other genes, like maybe genes for ear attachment, then we get these recombinant ones so that we have, the gene that used to be with one type of ear attachment is with another one. And because of this, we end up getting a lot of different variation in the gametes that are produced. So what we should write here is that crossing over, or in crossing over, genes get switched between adjacent sister chromatids. Now, because each of those sister chromatids is eventually going to end up in a different cell, whether they're two different sperm cells or two different ova, that means that those sperm cells or ova are going to be distinct from each other. They're going to have differences. This is one of the main reasons that all offspring do not look exactly the same, because we have this recombination of genes when we make gametes. Okay, this shows the same idea, except here we actually have all four sister chromatids. So this would be the tetrad right here. Okay, and the part where the crossing over occurs, where the switching actually occurs, is called a chiasma. Okay, now when we start out on a single homologous chromosome, the two sister chromatids should be identical. So here, they've used colors and capital letters to show that these two sister chromatids are identical. On this homologous chromosome, these two sister chromatids are identical. When they join up during prophase 1 and this crossing over happens, what we end up with is 4 distinct sister chromatids. Okay? This one is kind of like the yellow one was to begin with, and this one is like the blue one was to begin with, but now we have these recombinant ones. And so from the process of crossing over, we get 4 distinct sister chromatids. So this right here is the main reason that all 4 cells that are produced from meiosis are unique to each other. Because the thing is, this can happen on every chromosome. So this is just showing it on one chromosome, but in a human, all 22 autosomes can have crossing over. If you have 2 X chromosomes, there can be crossing over. Okay? There's not a whole lot of crossing over that happens between an X and a Y, because they are different chromosomes. But the other thing is, this can happen at any point. Right here, it happened between the I's and the J's, right? But there's nothing that's saying that in another time that this happened, it wouldn't happen between the K's and the L's, or between the A's and the B's, or whatever. So all along the entire chromosome, we can have crossing over points. And because every chromosome can cross over, there are literally billions of different ways that a person's DNA can get swapped around. So that means the likelihood of producing 2 identical sperm cells or 2 identical ova is really 1 in, I think it's like 5 billion. Yeah.
So is that how doppelgangers happen? Like, if someone identical to you, but it's related?
That's an interesting way you took that. I guess sort of. That really comes down to the fact that there's a lot of these genes that if they get switched around, we wouldn't really see a difference in 2 people. Okay? So, I mean, I'm going to say sort of. But what this really explains is the fact that you can have a family where a husband and a wife have 15 kids, and all those kids are different, right? They're not all going to be exactly the same because every sperm cell and every egg cell that is produced is going to have distinct DNA. And then if you think about it, when you're talking about 2 people having kids, you have to multiply that, those chances by 2, because even if you were to have 2 of the same cells that are produced, what are the likelihood that they'll find each other during reproduction? So identical twins happen differently. We're going to talk about identical twins on Monday when we come back. And identical twins come from the fact that a fertilized zygote splits after it's been fertilized. And we'll look at that. But there are things called half-identical twins. We'll talk about how that happens too. Okay. So that all happens in prophase 1. So prophase 1 is kind of the special part, the most special part of meiosis, because that's where this genetic recombination happens. And I think it would be good to write that term down. So let's put that here. So this is a form of genetic recombination. Now, the shorter the chromosomes, the less likely there's going to be distinct crossing over events. So you're more likely to have lots of different places where crossing over can occur on, say, chromosome 1 versus chromosome 22. And, of course, the fewer chromosomes you have, and here we're talking about species, so species that have fewer chromosomes, there's less genetic variation there because there's not as many chromosomes to cross over. Now, after prophase 1, the next stage is metaphase 1. So in metaphase 1, the tetrads line up in the middle of the cell. And the spindle fibers are going to attach to the centromeres, just like in mitosis. But in this case, it's a little bit different. Because we have a tetrad, one spindle fiber is going to attach to one homologous chromosome. The other one is going to attach to the other. And as we'll see in anaphase, what happens is the homologous chromosomes get separated. Okay, so just like metaphase in mitosis, we have things that are lining up in the middle. But just to make sure we understand the distinction, the next picture I'm going to show you, we're going to compare metaphase 1 of meiosis, which we're looking at right now, back to metaphase of mitosis and see the difference. A couple people finishing up writing. Let them get. Okay, now you can see in this picture that crossing over has occurred because this chromosome, which is mostly blue, has a red end right here, and this one, which is mostly red, has a blue end, and vice versa. Okay. And which chromosome ends up where is really just a matter of luck. Okay, there's just as much chance that this chromosome pair, this tetrad, could have lined up the other way with the red, well, the one that's mostly red pointed this way and the one that's mostly blue pointed this way. And when we compare that to these chromosomes, compare that to these chromosomes, Tate, then there is a whole bunch of different ways that these things could line up, and we call that independent assortment. We'll be talking about that a little bit later. So here, this is our meiosis 1 metaphase. So metaphase 1, here's metaphase of mitosis. Notice the difference. Yes, they're all lined up, but here we have each chromosome that has a spindle fiber attached to each side, and it's the sister chromatids that are going to separate. Here we have the tetrads, and each of these tetrads is going to pull apart, so we'll have a homologous chromosome going each way. Okay? If you remember yesterday, we wrote down the whole point of meiosis 1 is to separate homologous chromosomes. It'll be in meiosis 2 where we separate the sister chromatids. Okay? So, given two pictures, we should be able to tell, hey, this one's in meiosis 1, this one's in mitosis, because, we don't see the tetrads in the mitosis one. Okay, the next stage is anaphase. Anaphase is where the separation actually happens, so this is anaphase 1. And so here are the homologous chromosomes. Oh, dang it. Hard to type looking over your shoulder. Homologous chromosomes are separated. So just like in mitosis, these homologous chromosomes, the centromeres are going to kind of move themselves along. Remember, they don't get pulled in. They actually move along like trains on a train track. And they're going to move into separate ends so that they end up in different cells. Okay, again, to compare, this is meiosis 1. This is mitosis. Here we see an anaphase of meiosis 1. We still have a homologous chromosome here. The sister chromatids are still attached. Here it's the sister chromatids that have been pulled apart. Okay, and then the last stage of meiosis 1 is telophase 1. What sort of things do you think might happen in telophase 1? Well, we'll start to pinch together. So, yeah, we can put that. Cytoplasm pinches. Yeah, we see that. Okay, the nucleus reforms. What's the other thing that happens in telophase? spindle breaks down. So very similar to what happens in mitosis. The distinction here is the nucleus doesn't completely reform. Because this cell, or this, whatever you want to call it, this dyad cell, hasn't completely separated into two cells yet. It's got to go through a whole nother division because the sister chromatids still need to get pulled apart. So each of these chunks right here on either side now has to go through basically the steps of mitosis. So each of these is going to go through all the steps again. So the spindle, or the nucleus kind of starts to reform, and then it starts breaking down again as we move into prophase 2. So all of meiosis 2 follows the exact same steps as mitosis. This thing will break down, these will line up again, the sister chromatids will get pulled apart, and what we'll end up with then is we'll have 4 cells, each of which have one sister chromatid from each homologous pair. They happen in both. Yeah, so there's prophase 1, metaphase 1, and telophase 1, and then it goes right into prophase 2, metaphase 2, anaphase 2, and telophase 2. Okay, again, here's a distinction. In telophase 1, we see these things are still homologous chromosomes. Here it's just the sister chromatids. So we should be able to tell the differences between those. So this just kind of shows all the stages of meiosis 1 that we just talked about. So just to reiterate before we move on to meiosis 2. Okay, so in prophase, we get the tetrads. That's where crossing over occurs. In metaphase, those line up. And then in anaphase, they get pulled apart. And then I didn't show telophase up here because we just went over that. Okay, but looking at this kind of summarizes all of the things that we just talked about. Now, as far as meiosis 2 goes, we're not really going to go through every single stage because it's basically identical to mitosis. In fact, if you look at a cell, there's no way to tell visually that a cell is in, say, meiosis 2 versus mitosis. So if you look at, say, cells lined up in metaphase, you could easily mistake this for metaphase of mitosis. It really comes down to what types of cells are we looking at. If we're looking at the production of gametes, it has to be, this part right here has to be in metaphase 2. Now that's only if you're looking at a single cell, though. In most cases, what we're going to see is we have 2 cells next to each other that are in the exact same stage. And that's kind of a thing that you can use as a distinction of whether we're looking at meiosis or mitosis. Because these 2 cells, right, already went through all the stages of meiosis 1. And so it's showing them separately here, but really they're going to be kind of glomped together just like this through all of these stages. So these should really be connected at this point. They haven't truly separated yet. Cytokinesis does not come until after meiosis 2. So these 2 cells, we wouldn't even call them 2 cells. They're still 1 cell in kind of 2 different parts. They go through all the stages of meiosis 2, and then cytokinesis will happen to separate all 4 of the cells. Okay? But we don't really need to go through every one of these steps because it's exactly the same. We get the breakdown of the nucleus, the formation of the spindle. They line up. They get separated, the sister chromatids get separated, and then the nucleus starts to reform and the spindle breaks down. All the same steps as mitosis. The only real difference between meiosis 2 and mitosis is that in meiosis 2, there's half as many chromosomes. It's a haploid cell. But if you don't know how many chromosomes to count, it could be a little bit hard to tell. So we're going to write down meiosis 2 is like mitosis, but the cells are haploid. So whereas in mitosis, we would see 46 chromosomes in humans that are all lined up, and the sister chromatids are getting separated. Once we get to meiosis 2 and the production of either sperm or ova, we would only have 23 chromosomes there. And as we'll talk about down the line, in males, these have already determined which ones are going to be X and which ones are going to be Y. Because one of these cells got the X homologous chromosome, one of them got the Y, and then the sister chromatids for the Y will break apart. So we'll end up with 2 cells with a Y and 2 cells with an X. And whichever one of those makes it to the ova, that's the one that fertilizes it. Although there is a common misconception, the first sperm cell there is not the one that fertilizes it. In fact, it takes a whole bunch of sperm cells to help break down a barrier that is around the ova, and so it's really one of the latecomers that actually does the fertilization. So sometimes you hear that, oh, you know, you were the first sperm cell. No, that's not what happened. The first sperm cells did all the work. The one that fertilizes it is actually the slacker. Okay, the other thing that we have to be careful of, and I tried to say this yesterday, meiosis is not a cycle. So you can see I have that right here. I usually circle it, but since I can't circle it, I'll just indicate it right here. Meiosis, remember, we start with 1 cell, we end up with 4 cells. Those cells can't go through meiosis again because they've already become haploid. You can't break those down anymore. In meiosis, we go from, in humans, 46 chromosomes in each cell to 23 chromosomes in each cell. And so there would be no way for that cell to also go through. Yes. In mitosis, remember the daughter cells are clones of the original, so they're going to have 46 too. So I'm going to write this down. You can if you want. We've already kind of wrote in that. But. So this one kind of shows everything, right? Here's that 1 cell starting out. You can see the tetrads here. So this is going through meiosis 1. And then we split off. We have these 2 cells that are kind of together. They're going to go through meiosis 2. And we end up with 4 distinct cells here. Oh, I lied, Logan. We're not going to talk about twins till Tuesday. Monday we got to go over genetic variation. Okay, I'm going to shut off the lights here real quick so we can see these a little bit better. So these are some microscope pictures of actual cells going through the process of meiosis. So it's not the easiest to see here, but this is kind of prophase. Here, they're forming the tetrads. It's a little bit hard to see, but here you see the anaphase 1 where the homologous chromosomes are getting pulled apart. Okay, and it goes through the same process, and we end up with 4 distinct cells here. This is just before cytokinesis. Okay, so at this point, it will move on to cytokinesis. See if I can draw an arrow here. And that will separate all 4 cells. down a little bit. I don't know why my computer's so laggy today. Well, that was fun. This picture shows the process a little bit better. So here we have the parent cell. Again, because they're clumped up, it's a little hard to see, but this would be where the tetrads are all lined up. Okay? Here's after, now we consider this after the first chromosomal division. So in this case, we have the homologous chromosomes pulled to either side. Okay, and then we'll see that go through the same process, except here they're going to move in these directions to separate. So after the second division, and then here we have the 4 gametes produced after cytokinesis. All right, last thing we need to put down. Okay, gamete production, which, of course, the whole process is called meiosis, in females is called oogenesis. And in males is called spermatogenesis. Genesis means the beginning. So this is the beginning of the ova and the beginning of the sperm cells. Okay, now in human males, spermatogenesis is continuous from puberty, basically late into adulthood. I think Charlie Chaplin fathered children late into his 70s. And so, who's that? Ever heard of Charlie Chaplin? No. You guys are so sheltered. He was a silent film star, kind of physical comedian. Hitler stole that from him. Hitler ruined it. But, he had the little mustache, and anyway. But he had kids late into his 70s, and so in most males, that sperm production just keeps on going. Now, females are a little bit different. We're going to talk about this on another day more in depth, but just to kind of give you a preview, females are actually born with all of the ova that they're ever going to make kind of in a suspended animation. So in the mother's womb, little girls actually are already making ova, but they kind of get frozen, and then over time, after puberty happens, those eggs, I mean, when we say frozen, we don't mean cold, we just mean they're kind of stopped, right? And so the production of the eggs of the ova is stopped for a while, and then every month that will start up for a single one, and it will produce a single ova. It turns out that we don't actually get 4 cells when females make ova. You only get 1 actual cell. The other 3 things that are made can't be fertilized. But we'll talk about that down the road. No, there's still millions of them because of all the cells that got started. But every meiosis only produces one cell that can actually be fertilized. The other three are called polar bodies, and even though they have chromosomes in them, they can't be fertilized. They're not full cells. It really comes down to females put all of the stuff from the cells into one to make sure that one cell has enough ribosomes and all that kind of stuff to survive. And so it's kind of like, I don't know, stockpiling in one cell. Okay, that's all I have for you today. Okay, your homework for Monday is to do 6.1.